Hello, everybody, and welcome to the second part of Chapter 6, where we'll be talking about chemical senses. And when we talk about coding in the chemical senses, and we're really talking about taste and the sense of smell, there could be two different possible ways that we code different tastes and different smells. So one of them would be a labeled line principle, in which each receptor responds to a limited range of stimulus and sends a direct line to the brain. So each receptor is responsible for a specific either taste or smell. Or we could have a cross fiber pattern of recognition of taste and smell, in which each receptor responds to a wider range of stimuli. And then it's really a pattern of the different type of stimuli that allows us to know which scent or which taste we are experiencing. So let's start with taste and how that works. First of all, taste is really influenced very much by our sense of smell to give us the flavors of different foods. So you'll see we have really quite a limited number of taste receptors in our mouths, but we nonetheless can taste a wide variety of flavors like chocolate ice cream and pepperoni pizza and all of that. And really, the flavor of a food is quite influenced not only by taste, but also by our sense of smell. If you lose your sense of smell for some reason, taste is impaired. And in fact, people very often lose a lot of weight because food doesn't taste like much of anything, and so it's not very appetizing or appealing to eat. And in fact, you might have had this experience yourself if you had a cold and a very stuffy nose, and so you couldn't really smell, and you try to eat food and it tastes very bland and flavorless. It's because you're not using your sense of smell along with your sense of taste. Receptors for taste are really just modified skin cells, but they have excitable membranes like neurons, and they can release neurotransmitters. Taste receptors are replaced every 10 to 14 days, and that's very important because taste receptors on your tongue get damaged, right? You eat some hot soup and burn your tongue. Well, if you damage those taste receptors, you don't want that to be a permanent problem. Or you eat a sharp, crunchy chip and also damage some taste receptors. Again, you don't want that to be permanent damage. So because our mouth is such a, let's call it a dangerous place for taste receptors, we're lucky that they are replaced about every 2 weeks. So we have these kind of bumpy structures on our tongue, the papilla. Those are just ways to basically increase the surface area on your tongue. And your taste buds, you can see these little blue taste buds, are contained on these kind of bump-like structures. There may be 10, up to 50, sorry, I've got a bunch of numbers there. But a number of different taste buds. Most of them are located along the outside of the tongue in humans, and not really many taste buds towards the center of your tongue. Taste buds and your sense of taste undergoes adaptation, just like we saw with the sense of we talked about when we began to talk about sensory processing, in that if you repeat a particular stimulus, you don't taste it as much. So the first time you taste something sour, you taste it quite a lot. And then if you keep tasting that same sour thing over and over, it doesn't taste as sour anymore. The first time you taste something very salty, again, you get that very strong sense of salty flavor. And then if you keep eating that same thing over and over, keep having that same salty flavor, it tastes less and less salty. That's the idea of adaptation. So the taste buds are embedded here. And extend out these microvilli right here, these little like little extensions that contact chemicals that we call tastants, things that you can taste. Um, and there are at least 5 different kinds of taste receptors. We know we've got sweet, bitter, salty, sour. Those were the 4 that we thought existed for a very long time. We've recently added a 5th taste receptor, umami, which is a flavor, of kind of a meaty flavor, and basically seems to respond to glutamate. And each of the receptors work differently. The salty taste receptor is probably the easiest to understand because it simply permits sodium ions to cross the membrane. So sodium ions just enter the sodium receptor, and what happens when sodium ions enter a neuron, right? The cell starts to depolarize, hits the threshold, and results in an action potential. So the saltiness receptors are really the simplest to understand, just allowing sodium to cross the membrane and lead to an action potential where you then taste salt. Sourness receptors seem to close potassium channels when an acid binds to the receptors, and those acids release hydrogen ions. Hydrogen is positive, and that seems to be what allows us to taste that sour taste. They all have the sour taste cells all seem to contain the same type of ion channel that allows an influx of protons. Protons are positive, or these hydrogen plus protons, which then depolarizes the cell and again results in an action potential. The other receptors, sweetness, bitterness, umami, seem to activate a G protein and a second messenger system, which is a lot more complicated, to allow us to taste these different tastes. Interestingly, genetic factors and hormones can account for some of our differences in taste and taste sensitivity. Some people are very particular in what they taste, and they seem to taste things much stronger. In fact, there are people called super tasters that are highly sensitive to all tastes, and they seem to have a higher number of taste receptors. And these super tasters may crave more flavorful dishes because it really is it's such a much stronger sensation of taste for them. And then they are at risk for obesity again because food tastes so much more strong and in some cases much more delicious, and so they're more able at risk for overeating. So taste is also called the gustatory system. It extends from the tongue, then to the brain stem. Again, goes through the thalamus. Remember, the thalamus is the sensory relay station, and ultimately to the somatosensory cortex. How do we taste these different tastes? The brain may simply monitor just which specific axons are active to determine which tastes are present. So this may be more of a labeled line system, that if you're tasting something sweet, those sweet receptors are sending a message, an axon, basically through to the brainstem, the thalamus, and the somatosensory cortex, and registering, okay, you've tasted something sweet. And then if you have tasting a salty flavor, again, the salty receptors would then be sending a message to the brain saying, oh, I'm tasting something salty. There is some evidence for this being labeled line versus a cross fiber pattern, because if you damage one type of taste receptor or inactivate it with a chemical, the other 4 tastes remain totally intact. So those are still able to send their message directly to the brain, it seems, and only you're getting a deactivation in just that one specific taste receptor. Whereas if this was a pattern type of coding system, if you inactivated one type of taste receptor, you would tend to see changes perhaps in all the other tastes, because that taste receptor would be important in the pattern of active fibers for all the different taste sensations. Various areas of the brain are responsible for processing taste information. Each hemisphere of the cortex is also responsive to the ipsilateral side of the tongue, same side of the tongue. So this is the one sensory modality that goes to the same side. And then primary taste cortex in the brain is called the insula. So that's where information is first sent to the cortex, to the insula. Olfaction, now we're at the sense of smell. It's the most subject to adaptation than any of our other senses. So again, when you smell a particular scent for a prolonged period of time, you almost can't smell it anymore because of our sense of because of our adaptation to that particular smell. Olfactory cells line a membrane in the back of your nasal passage called the olfactory epithelium. And that's where the neurons that are responsible for smell are. Odorants are the inhaled molecules, the chemical molecules that interact with olfactory receptors, proteins on the dendrites. So here are the olfactory cells, and you can see that the olfactory neurons are a little bit different from neurons in the brain, but they also have these down here. Let's see the dendrites, kind of see them here, or we call them cilia, but they're basically like dendrites reaching out to get the chemical odorants that come in through your nose. Then they all come together in this dendritic knob. You've got your receptor cell here with the nucleus, and then sending the axons back into other parts of the brain. There are an incredibly diverse number of receptor subtypes for the sense of olfaction. We smell so many different scents, and there are many, many different olfactory receptors, in fact, probably more than we even know at this point. Also, these neurons or these receptors here survive for a little over a month and then they're replaced. So same as taste, right? If you smell some kind of toxic chemical and end up damaging some of these sensory receptors, they will come back, which is very important. So the olfactory receptors, like I said, are located on the cilia and extend from the cell body into the nasal passages, right in here. I've already said the rapid adaptation to scent. Each receptor sends impulses then to the olfactory bulb. Okay, so here's the olfactory bulb right here. And as I said, vertebrates have hundreds of olfactory receptors to process such a wide variety of airborne chemicals that we can smell. Axons then, after they go to the olfactory bulb, coding is determined by which part of the olfactory bulb is excited. And it seems to be that then the axons send these messages to the cerebral cortex, and the cerebral cortex codes your sense of smell by what part or what location of the cortex is activated. The cerebral cortex connects to other areas of the brain that control feeding and reproduction, which are both highly influenced by smell. And so it's interesting that there's a lot of connections between our sense of smell, areas of the brain that process our sense of smell, along with feeding and reproductive behaviors. Olfactory receptors can be permanently damaged and permanently impaired after real massive damage. So this often happens when someone has a really big head injury right to the front part of their brain, maybe a car accident where the front of your head, your forehead, think about kind of the bridge of your nose, that's where the receptors all kind of go through in the back there. And so those can get damaged. And with massive damage, they are permanently impaired. We've got the word anosmia that refers to a general lack of olfaction, general lack of smell, and a specific anosmia, that's a specific inability to smell a particular type of chemical or a particular smell. All right, so structurally different but located near the olfactory receptors in the nose is this vomeronasal organ that has receptors that respond to pheromones. So not to chemicals that are produced in the environment, but instead particular chemicals that are released by animals that affect sexual behavior of other animals, and we're including humans in this as well. This vomeronasal organ is much smaller in humans than it is in other animals whose sexual behavior is much more controlled by pheromones. But in humans, we also respond to skin secretions, these pheromones, and these pheromones cause increased activity in the hypothalamus. Hypothalamus is an area of the brain that's important for sexual behavior. And we see there have been different studies that show that women who spend a lot of time together, like live in dormitories together, and they're releasing pheromones all the time, eventually their menstrual cycles will synchronize and happen at the same time. It's also shown that women who are in intimate, close relationships with men over time will have an increased regularity of their menstrual cycle. Again, just ways that these pheromones really impact our sexual behaviors. All right, and that's the end of this chapter. Thank you very much.